Good.
Great. Good morning. Thank you all for coming, and thanks to the network for inviting me and for your support. So I don't really need to, I guess, provide this material, but it's here so you all know that strong social support systems enhance the quality of life, they improve resiliency, and they reduce the risk of morbidity and mortality from a whole variety of diseases in marriage, companionship, and association in couples of all types, and the social support they provide have specifically been recognized as contributing to improved survival in patients with cancer. And much of the literature on this area says that social support is because who's ever providing the social support can cook your meals, help you around the house, go to the pharmacy, pick up your prescriptions, get you to your doctor's appointment, and because of observations that we had in our mouse breeding colony where they don't drive and they don't go to the supermarket and so on, we figured, well, there's obviously something biological, and had I been at this meeting before, I would know from all of you that you already know a lot about the biological aspects, but at any rate, we thought that animal models are needed to better evaluate the biological aspects of social support systems, and we do a lot of work on behavior like diet control and physical activity on the APC-min mouse model. The APC-min mouse model is adenomatous polyposis coli, and any mouse that carries that gene will get intestinal neoplasia. MIN stands for multiple intestinal neoplasia, and essentially 100% of the mice that carry that gene will ultimately get multiple intestinal neoplasms and succumb to those mice. And because of observations that we had in our breeding colony, but that we never really tested, so we formally hypothesized that mixed gender social support groups would provide greater survival benefits for these APC-min mice with their spontaneous intestinal tumors. And we wanted to compare that mixed gender support system to mice with the same mutation that were incubated in social isolation, and we also questioned whether mixed gender grouping would have the same or different effects than same gender grouping. And so here is our setup. We compared the effects of 3 different social structures on survival, on tumor growth, and on circulating factors in male APC-min mice on a C57 black 6 background. And we picked 2 time points to do all our comparison. One was when the mice were all 90 days old. So that meant that all the mice that we looked at, with whatever social support system or whatever system we used, they were all the same age, and that's at a time before they showed any external signs of disease. We know the tumor is growing inside of them, but there's absolutely no visible external sign. And then we also looked at the time that the mice were moribund and ready to die. And each one of these groups had at least 20 mice in them, so we're talking about the results on 120 mice today. So the 3 groups that we use were, the first one is social isolated housing, where we have one male APC-min mouse from the time it's weaned. It's incubated alone. We decide the bottom, let's see, the mixed gender grouping, which is the ultimate experiment that we wanted to do, was 1 male APC mouse with 2 B6 female mice. That's our usual breeding arrangement. And then to see what effect same gender housing would have, we put 1 male mouse with the tumor gene in with 2 normal B6 mouse, and then, as I said, we measured survival and other factors, and then we looked at circulating factors, and we used two panels, one by Myriad rodent biomarkers. So this was a panel that they put together that really has no predetermination as to whether it should have any psychosocial impact, and ampersand bioscience, another smaller panel. And here's our survival curves. The blue curve is the mixed gender housing. The green curve is the isolated housing, and the red curve is the same gender housing. And you can see that the mixed gender housing is clearly more effective than the same gender than the isolated housing. And if you look at the 90-day time point, which 90 days is right, I can't get this to move straight, but anyway, 90 days is right here, which is essentially before any of the mice are dying, and you can see that the green bars, which represents the total number of intestinal tumors, and it's a good indication of tumor mass, is highest at this 90-day time point in the social isolated mice, and least smallest mass in the animals that were in the mixed gender housing. What we found very interesting was if we waited till the animals died, which was again at different time points, that the mixed gender, that the animals who had the mixed gender housing, they had the highest tumor mass. So that means that the effect of the mixed gender environment allows these animals to survive longer with more tumor than the ones that were socially isolated. So just to show you just a few of the numbers here, the top three bars are the mice that we sacrificed at 90 days, and these are the ones that we let go to their spontaneous demise. And here you see the mixed gender housing survived for 164 days, which compared to 124 days for the social isolated mouse, with really statistically significant difference. And the rest of the numbers probably aren't worth dealing with on this. So then we looked, as I told you, at 50-some circulating factors, and this is an example of just one, the C-reactive protein. And so what you see here in blue are the range of values for the mixed gender mice. Here are the same gender mice, and here are the isolated mice. And the asterisk here indicates that the same gender mice at that the social isolated mice at 90 days, again before they showed any signs of disorder, were, can I get this to go back? Were significantly elevated compared to the mixed gender, or alternatively, you could say that the mixed gender were, in fact, lower than the social isolated. We then looked at these same factors just before the mice died, when they were moribund, and the significance indicators here compare this number to this number, or this number to that number, or this number to that number, and you can see that all of these decreased relative to what they were at 90 days, but in this whole panel that we did, we saw nothing at the time of death, no difference in any of the factors at the time of death, which in retrospect was probably not too surprising. So here is more data than anybody can look at, but again, here is the CRP, and you see that this is elevated. Here's eotaxin, which is decreased. If you look at a number of other factors at the 90-day time point, we really don't see any difference. Here is macrophage-derived chemokine, which is significantly decreased in the social isolated animals, and here's macrophage inflammatory protein 3 beta. Some of you people who are the immunologists probably know more about this than I do, but so you see that the effect of isolation versus mixed gender housing is really having a significant effect on some of these circulating mediators. And here is another group, and you can see that epidermal growth factor is decreased, and I think that's the most significant one on this panel. And here is corticosterone, which was elevated under conditions of social isolation compared to mixed gender housing, and I think that was the only one that's really significant on this panel. Now, another thing that we did is we looked at insulin-like growth factor 1 and insulin growth factor binding protein 3. So insulin growth factor 1 is a cancer promoter, and insulin growth factor binding protein 3 more or less neutralizes it from being able to act on tumor cells. And what we did is we subtracted the insulin growth factor binding protein from the insulin growth factor 1 that we measured in the circulation. And you can see, and if it's above the line here, there's a waterfall plot of each one of our mice. And you can see here that if it's above the line, it means that there's more free circulating insulin-like growth factor 1. So these mice at 90 days are more prone to tumor growth, if for no other reason than they have more of this free circulating IGF-1. So this summarizes our results that in male C57 black 6J mice with a genetic predisposition to intestinal cancer, they showed differences in disease progression and survival associated with different social structures. So mixed gender housing improved survival by 32% compared to social isolated housing, and by 13% if they were the same gender. Mixed gender housing was associated with greater survival, and the social system was associated with same tumor growth at 90-day time point, but mixed gender housing compared to social isolated housing animals had greater tumor numbers at the time of their cancer-related deaths. At the pre-morbid time point, that's the 90-day time point, we found an increase in the isolated versus the mixed gender housing in corticosterone and C-reactive protein. We found a decrease in IGF-BP3, which, as I showed you, the subtraction led to a consequent increase in free IGF-1. There was a decrease in epidermal growth factor, a decrease in adiponectin, a decrease in macrophage-derived chemokine, and a decrease in macrophage inflammatory protein. And again, the social structure had no significant impact on circulating factors by time of cancer-related deaths. One of the reasons that we did this, that we did these experiments, is that we do a lot of work with exercise in the mice, and there's a lot of suggestion that there should be, that people who have cancer will really benefit from exercise, from yoga, from increased sleep. There are all sorts of behavioral modifications that are suggested. The question is, how much? Some people say that, you know, if you exercise 15, 20 minutes a day, that's fine. And other people say if you really want to improve your survival, you need to hike across the Alps, you need to be a Tour de France bicycler runner. And what, you know, one way to look at this is to measure the impact on survival. That's obviously in people, that's an extremely difficult parameter to measure. So what we're interested in is identifying parameters in the mice that tell us that this is associated with a better survival than some of these stress factors or loneliness, and see whether they apply to some of the behavioral abnormalities. So if we take a group of people who are doing low exercise and look at some of these parameters that we see in the mice, and if they're low, can we design a study to increase their exercise till they get to the level that are increased with improved survival. So that's our goal with this. So that just, that summarizes what I just said. Mixed gender housing is good for you. And by the way, this premise that I just gave you, it would be nice if you could take all your cancer patients who are single or who don't have group activities and say, well, go find somebody to live with, or but that's hard. So the question is, do you tell those people to exercise every day? Do you tell them to exercise alone or in groups? And there's all sorts of, potential perturbations of this, and it would be nice to have an endpoint to measure other than survival, which is actually, from our viewpoint, the ultimate endpoint. So thank you for being such great sponsors and listeners.